Hello friends and my dear students. Today we are going to see the part of genetic material, that is the structure of prokaryotic chromosomes, and under that we will see the E. coli, Escherichia coli as a genetic model, as a microorganisms under the prokaryotic chromosomes. So friends and dear students, as you know, the chromosomes of prokaryotic and eukaryotics. Why we are going to study the prokaryotic chromosomes? Because uh, prokaryotic chromosomes is very important as far as in the concerns of research, and we can use such chromosomes under laboratory level by changing its strains variety, and due to which we can produce a number of new genetical varieties of microorganism as well, the hybrid varieties of plants, crops, whatever it may be. On the basis of that, let us see the prokaryotic chromosomes. The prokaryotic chromosome is also called as primitive nucleus because it is immature, or it is initial stage of the nucleus in case of prokaryotics, and there is no complete form nucleus, because whatever the it is nucleus, or whatever the chromosomes which is in thread like structure, it is dispersed within the cells, which is dispersed within the cytoplasm of the bacterial cells. It is not well developed. It means that there is no chromosomes, there is no chromosome, sorry, there is no nuclear membrane or other nuclear parts within that. Much of the informations about the structure of DNA has comes from the studies of prokaryotics, because they are less complex. Prokaryotic chromosome is less complex as compared to eukaryotic chromosomes, as as compared to eukaryotic chromosomes regarding the genetically and biochemical structurally. Prokaryotics are monoploids. Monoploids means they have only one set of genes, and there is only one copy of genomes, because in bacteria and viruses there is the presence of DNA or either RNA. It means that there is the single set of genes is stored in the DNA form or RNA forms. It is prokaryotic chromosomes or genomes are amplified or exemplified by the E. coli chromosomes. E. coli, because E. coli organism is an experimental genetically observed organism, because for the first time the scientists they observed the chromosome of Escherichia coli, and whatever the study which is based on the E. coli, and the bulk of the DNA in E. coli cells consists of a single closed circular DNA. Whatever the DNA present in the chromosomes, which is a single and closed circular forms, and the length of such DNA, which is near about 4.6 million base pairs. This much amount of base pairs are present within the DNA of E. coli. The DNA is packaged into a region of the cell is known as the nucleoid. It means that DNA is generally fixed, or DNA is generally attached within the cell. That attached part, or where the regions, that region is called as nucleoid. DNA domains. It means the detailed structure of prokaryotic chromosomes. Uh, when the DNA of prokaryotic isolated by using isolation process from other proteins, and that isolated DNA when observed under the electron microscope, it looks like this type of structure. You just see the structure, that is the central part of that central part of the DNA that generally attaches to that is called as nucleides, and the DNA consists of 50 to 100 domains or loops. It means that it is supercoiled, because DNA is a double-stranded helical structure. It is supercoiled chromosomes. They are found around the centers, and near about 50 to 100 loops, the ends of which are by binding to a structure which is probably consist of proteins attached to a part of the cell membrane. The loops are about 50 to 100 kb in size. It is that much size of the chromosomes. The supercoiling of the DNA, the E. coli chromosomes as a whole is negatively supercoiled. Negatively supercoiled means it is a one loop is further supercoiled. One fold is further. What happens? It undergo into the folding. That is, we can say supercoiled. There is more evidences that the individual domains may be supercoiled independently. The electron micrograph indicates that some domains may not be supercoiled, perhaps because the DNA has becomes broken in one strand or where other domains clearly do contain supercoils. The attachment of the DNA to the protein's membrane scarf may act as barrier to rotation of the DNA such that the domains may be topologically independent. Means there's place where it binds with each other's DNA binding proteins. There are number of proteins they are binded with DNA surrounding that within the cell, because the most abundant form of the protein, that is the HU, which is small basic, that is the positively charged proteins, because each protein has a specific charge, positive or negative, and which is present on that. Apart from that, it binds DNA non-specifically. Some proteins they are bind specifically, and some proteins they are binds non-specifically. And so DNA around the proteins, the HNAs, formerly known as protein H1, it means that number of proteins are present, and each protein has a specific activity, specific role. On the basis of that, they also named a specific name. The monomeric natural proteins, which also binds the DNA non-specifically in terms of sequence. It means that specifically they are attached to the DNA, which is essential for the packaging of the DNA into the nucleotide and is stabilized and constricting the supercoiling of the chromosomes. Half of this constrained as a permanent wrapping of DNA around proteins such as HU. Only about half of the supercoiling is constrained. It means the amount of proteins is, it means HU protein is much more as compared to other proteins in the concerns with the DNA attachments. And apart from that, RNA polymerase is the enzyme which each and every proteins enzyme, they are made up of proteins, but all proteins are not enzymes like that. And mRNA molecule site specific DNA binds proteins such as integration host factor or homology HU, which binds to a specific DNA sequences and bend DNA through. It means the number of proteins binds and number of enzymes, number of molecules, because it is generally based on the specific. So this is the figure. This is the diagram of the structure of the E. coli chromosomes. It initially it looks like it is a round spherical. You just see the structure of that chromosomes. It generally looks like supercoiled structures, and it is round spherical structures generally. But afterwards, what happens? There is the changes. Changes means it further undergo into the folding, and their number of loops are there forms. After that, each loops has number of loops. It means supercoiled loops. And when supercoiled chromosomes of E. coli when further undergo into two parts on the basis of RNase digestion enzyme and DNase, and they have a specific RNA cleaved and nicked DNA, and that looks like supercoiling structures. Okay, this is all about the prokaryotic chromosomes. It means that due to the supercoiling and due to the round spherical natures of the chromosomes of prokaryotes, they are very simple structures compared to prokaryotics, and it is very easy for handling in the laboratory levels. So friends, with the end of such point, the genetic material, the unit is over, and I will send you the Xerox copy of such topic on the WhatsApp group. So after that we'll start the next unit, the prokaryotic replications, the prokaryotic chromosome DNA replications. Till then, bye.